is the research view, and we will be joined by Shira and Piotr, who are co-leaders of the Children's Rare Disease Collaborative at Boston Children's. Piotr and Shira, do you want to come up?
Thank you.
What the CRDC does, how it was established, and what you kind of view as its key accomplishments to date.
Yeah, thank you. So I think Jamie and David gave us a pretty nice overview of how the community is being built around the patient families. I guess our task is to kind of build similar community within Boston Children's Hospital between scientists. I don't know how much you know about how the academic institutions, pediatric institutions are run. We typically have a number of very big labs who are kind of completely engaged in genomics, but it's very few of them. And the challenge is to kind of spread it to other scientists, physician scientists who work with rare diseases, work with patients, and have been, very carefully building the cohorts, doing phenotypic analysis, how to bring genomic abilities to them to allow them to basically bring those diagnostic odysseys to the finish line. That's been really, really challenging. And so CRDC was basically established in 2018, and the goal was to do just that, to bring genomics to the physician scientists outside of these big groups. And what we did as part of the initiative, which was at the time led by David Williams, who was our CSO, was to do kind of across the board at PCH, not on the clinical, but on the research end, to make genomic sequencing available to anybody who really needed it and had the good information, was working with the physician scientists already at the institutions. So our goals were to first ensure that the data that is collected through CRDC can be broadly shared and make available across the institution to other scientists, potentially computational biologists who want to take a look at it and try to help us go a little bit deeper with the data analysis, but also on the flip side, not just collected for the research purpose, to be able to share it to support clinical care at PCH. So it's part of building this framework. We ensure that patient-led identifiers are collected, and then in cases when the genomic data can help, we can reflect that data to patient families and help to bring those diagnostic odysseys to the end. I don't know, Shira, if you have anything else to add.
I actually wanted to show this slide, which, okay, came up here. Here we are. Okay, so this is the investigators who are involved in the Children's Rare Disease Collaborative, and you can see it's really a big team effort. And yeah, this has led to a lot of, patients receiving those clinical diagnoses, research findings that are going to translate into clinical care in the future, as well as a number of, further research that's happening in animal models and translational work.
Great. So this is obviously a very large team working on this effort. How does the CRDC go about choosing its sort of focus areas?
So that's really driven by the investigators at the institution. And so, we had a call for proposals. We actually have recurring call for proposals by research investigators across the institution. And we're really, it's kind of a hybrid model where the investigators are working to get research funding to do some of the patient phenotyping and collect information on patient history. And then we're helping them on, once the patient is consented, on doing the sequencing and then the interpretation and analysis. So yeah, as Piotr mentioned, the original process in 2018 really identified a huge number of patients at Boston Children's Hospital who would benefit from this kind of genetic testing.
And how does CRDC work with the patient families and sort of leverage the information that it has to best deliver for the patient families? And just what is the interaction like, if you could speak to that a little bit?
So I think the primary interaction is not through us, because we're really collaborative to bring scientists together. And there's been kind of traditionally, all of this is done through our physician scientists. They work directly with the families, communicate with them. So we try to facilitate this communication, and kind of try to take a data-driven approach as much as possible. For example, this year we collected the list of all the patient advocacy groups that potentially overlap with our cohorts, and we share it with our investigators. Just kind of encourage them to work with the community more broadly than they already are. right? And also kind of across the entire collaborative with over 100 scientists, kind of encourage them to go deeper and potentially, bring them to the events that we're trying to, maybe you can mention the symposium we're trying to put in place in
Here's a slide. Oh,
in April. It'll be the first time we will do it.
yeah, so we're organizing a symposium at Boston Children's Hospital in April, and we're bringing researchers, patient families, patient advocacy groups all together for the day to kind of look at different perspectives, like we're doing here, on rare disease.
Yeah, and another effort we started. So this was actually one of the recommendations from our external advisory board about a year ago. Number one thing was to engage more with the patient families across the entire collaborative. Another thing, we're completely redoing our website. It's going to be quite informative about, for each disease, we're going to have disease-specific websites. So it all has been drafted. As long as we can get through marketing and put it out, I think we should be able to do it fairly soon. But I think the kind of bottom line, we're not doing enough. We can do more. But I think relationships which are in place between our physician scientists and the families can go pretty deeply. They do go pretty deeply.
So what would you say have been your biggest challenges to date? I know there's a lot of aspects that we've talked about so far, from the research to sort of building more of the relationships between the physicians and the patients and data sharing. What in all that would you say has been sort of the biggest challenges or the biggest challenge to your work?
Obviously, I believe number one is always funding, right? We're trying to stretch it. But Shira mentioned RFP process. We've selected cohorts. More people want to get on board. From the clinical side of the house, I think it's pretty hard to get clinical sequencing done right now. From our end, we want to go deeper and deeper. We're doing whole exomes, whole genomes. We're doing long-read sequencing right now. We want to, we bring new modalities with the data analysis. There's kind of infinite number of things we can do. And very nimble approach, right? Because we don't go kind of across everybody that comes through the door. We just focus on the kids when we think this could be most promising. And when we do those more deeper modes of analysis, it's always kind of going after negative. Whole exomes will go to genomes. If the whole genome is negative, we can still go to long-read sequencing. The kind of finding support and funding to be able to do it. Shira mentioned we're being some funding from the research group, some funding from the hospital, but between the fairly small staff that we have to support the whole initiative, that is basically just under 10 people making it go together. I think more resources, more funding could improve.
Yeah, and to add to that, like the CRDC is really sequencing patients who maybe have milder forms of disease that wouldn't be eligible to get clinical sequencing happening. And the research that's coming out of the CRDC from various cohorts is showing that a lot of these patients with more mild phenotypes actually do have a genetic underpinning for their disease that's able to be detected, right? So there are a lot more people, to Piotr's point, that can benefit from this.
One thing, when we started 5 years ago, we maybe have like 110, maybe 5 physicians at BCH who really understood genomics, and could pretty much work with any patients. I think we expanded this group to over 50 people who kind of move left and right and then have pretty advanced discussion. So growing this community, because to work, to Jamie's point, to work with this level of data, it takes a lot of effort, a lot of understanding, and it's just much more time consuming when you work with a single patient, single variant, this level of information we collect to be able to fully utilize it.
Yeah. Is there, looking at this list, this is a tremendous amount of focuses that you have. Is there a particular project or particular focus that either of you have found particularly rewarding, or just some example of your work? I know we talked about challenges, but can either of you think of some example of a project that went really well and was particularly fulfilling? Just any kind of example of a success that is particularly memorable for either of you.
Don't you have the slide from Anne?
Oh yeah, this one, yeah.
You want to talk to this? Or?
Yeah,
okay. Yeah, so I think this is a really good example, actually. This is from one of the researchers at Boston Children's Hospital, Ann Paduri. And basically, 2 siblings come into the hospital many years apart. The first one is a patient who gets treated with standard of care, and the outcome is a tracheotomy and G-tooth dependence. His sister comes in many years later with a similar phenotype, but this is after the CRDC had been established, and she was able to be enrolled in the research study, get that genetic diagnosis, and then also participate in further research to confirm that genetic diagnosis. And because the care was able to be enacted so much earlier, the outcome is substantially better. So I think that's one example, but there's countless stories. We actually, to Piotr's point about the website, we have, I think, 25 patient stories that are going to be coming up on the website for each of the different cohorts, kind of each describing a similar story to this.
Great. Yeah, it's amazing just in the few years that you've been established, the amount of projects that you've already undertaken and the amount of impact.
But also, like maybe, to David's point, I mean, we can go pretty well right now to at least to this point when we have the diagnosis and kind of shorten the diagnostic odyssey. To get to cures, it's just completely different trajectory. It's much harder. I think we do it quite well at BCH with Tim Yu, David Williams, and David Bauer. I mean, there is definitely leading in some spaces, but to scale it up and kind of do it across the board, it's a very tedious and time-consuming effort. So I think from our end, it's really kind of interesting to see examples when we actually use the genomic data to like with gene therapies, we can use it in kind of the different modalities right now to predict off-target effects and things of that nature. So we can see genomic data going just beyond this diagnostic odyssey and helping to develop therapeutics. I mean, it's still very early days, but definitely kind of one of the most satisfying parts of what we do.
So I'm going to shift the discussion a little bit. One thing that I want to talk about is, I know one of the unique things about the CRDC is that you have industry partnerships. Could you tell us a little bit about those? I know like you have one with Clinithink, for example, and Gene Diagnostics, Pacific Biosciences. Could you tell us a little bit about what the focuses of those are and kind of how, just at a high level, you might structure those?
Yeah, so each of them, I mean, these partnerships happen kind of bidirectionally and organically, right? So for example, Clinithink, there's a collaboration was started by an investigator at Boston Children's Hospital, which turned into a publication kind of analyzing how the Clinithink system is able to look at the clinical notes and pull from the clinical notes human phenotype ontology terms, which are really helpful in the genetic analysis. And can change over time, right? When a research team is looking at a patient, they look at the patient when the patient is enrolled. They might look at a patient on regular intervals because they're relooking at analysis. But with a tool like Clinithink, we're able to kind of in real time get those human phenotype term changes as the patient is being seen at the hospital. And so it started, with a collaboration with an investigator, Dr. Alan Beggs, to really assess, well, how does this match up compared to work that is being done manually, right? And that turned into a paper. And so, I mean, this is kind of a theme. I think like we collaborate with partners in industry to really have scientific outcomes from the work. So that can be seen in this Clinithink work. We also have there's a paper coming out that's in, I think, bioRxiv right now, but it's in the process of getting published with PacBio, looking at like long read sequencing for congenital sensory neural hearing loss, which also led to a white paper. So like really what we see is, collaborations where the company is invested, where we're invested, and we're working closely to achieve a scientific goal and better understand the underlying disease.
So do those usually, I want to ask a little bit more about how those are established. Is it usually an investigator who just has an interest in working with a particular partner, and then, the partnership just kind of develops organically after you sort of negotiate the terms? Or how are most of your partnerships with industry partners established?
I think it's really heterogeneous. So like the Clinithink example did start with a specific investigator, but the work with PacBio that we did actually was kind of coordinated at a CRDC level. And so that has many different investigators who are each contributing samples and contributing analysis. And then, we also work really closely with GeneDx. Which has been a work that's kind of morphed over time, but, you know, we do our sequencing with them. They're kind of our preferred vendor for like exome genome sequencing. But then they are also doing a lot of the sequencing on the clinical side of the hospital. They happen to also provide a software platform that we find really helpful. So yeah, it kind of runs the gamut.
Mm-hmm. So I know a lot of the attendees here are on more of the industry side. Is there anything that you would say from your experience sort of makes for a good partnership, or that's been challenging in your partnerships? Like, what would you say makes a good partner, and what have been some obstacles that you've kind of had to work through?
Yeah, I mean, in some cases, like working with companies on a longer term basis helps. Like GeneDx was this good example. I mean, we work with them right now pretty much since we started CRDC 2019. And like the longer way, like new projects basically keep emerging, right? We started initially with focus on the bioinformatics and then sequencing, and new opportunities keep happening. So like for any collaboration, when you work longer, you know, build up the foundation, the things will happen, right? Then it's always easier to kind of communicate with people that you already work with. I mean, obviously, you know, we try to find companies that are leading, like finding new things. We are research, non-clinical projects, so finding things which are on the forefront, and working to kind of define the pathway for the rest of the pediatric community. I mean, those things are always of interest to us. And, you know, our physician-in-chief, Wendy Chang, I mean, she's been very, you know, amazing since she started last year to create new opportunities. We interested to talk about it. I would say we're interested in things which are on the forefront and kind of ahead of where we are today. Anything else to?
I agree with that. I think just from a practical perspective, also when both parties, so like BCH and the industry partner, are really invested, I think that's what's driven the most science and made for the best partnerships as well.
Yeah. And I know one thing we touched on a little bit earlier was data sharing, and I know it's really a goal of the CRDC to just make data that you generate available. So to the extent that you're able, are you able to tell us how you go about, or what is sort of the philosophy around sharing the data that might be generated around these partnerships? Because I know just from working on our side of the industry, that's always a little bit of a sensitive area of sometimes academic partners really have a motivation to publish, whereas sometimes the industry partners are, you know, there might be some reasons to hesitate for patent coverage, et cetera. So how do you balance what might be a competing interest there, and how do you generally, what is the general approach that you generally try to take on sharing data that might come out of your partnerships?
So maybe, you know, one foundational things we did when we set up CRDC, all the data is from get-go consented for sharing, including potential industry partners, right? That as long as the data will support the patients down the road, we have that in the consenting. So that's kind of foundation, we have it, and that's been very supportive. I think the second layer is kind of our institutional discussion, right? Because we have people with different views, you know, everybody, you know, we have a committee. So when the data is to be shared with industry, and that's probably one of the biggest challenges. Like early on when we started, a number of companies offered us free sequencing, right? But when you look in footprint, like, yeah, we have to give you all the data, et cetera, right? So the kind of those agreements, when we give you all the data, no question asked, are not going to work with BCH, right? It has to be really thoughtfully designed project, you know, collaboration, et cetera. And then we have this internal process. If it truly makes sense for us and for the patients, the patient families, I think there is a good chance that it might happen. But there's a very kind of a tedious, long, kind of both legal as well as compliance conversation that would have to happen. But before we started, it wasn't the case, because the data from any cohort was specifically consented just for the specific cohort, and every single research study at BCH that would use genomics would have a different consenting protocol. So when we even trying to make sense out of it, combining data from before 2019 times, it's almost impossible to work with it, even in the research universe.
Yeah, even inside of our hospital, trying to run a query, for example, how many patients are there with a variant in whatever gene across all of the data in the hospital prior to the CRDC, was a challenge.
And I know you have collaborations outside of industry too. You have collaborations with other research organizations like Matchmaker Exchange and the International Children's Health Partnership. Could you tell us a little bit about those two and how those are alike and different from your industry partnerships?
Yeah, so I think the good example is the IPCHIP consortium that we co-founded a couple of years ago, and this is with Sick Kids, Gosh, and Murdoch in Australia. And I think data sharing challenge, again, number one, because now we have to share data across continents and different countries, right? So we still haven't sorted it out. We're looking at different technologies to kind of build a single platform where we can in a compliant way work on the data as the consortium, but we managed to bring all the physicians from all the sides and kind of, more enthusiasm for working with data. You expand the number of patients involved in a particular study. So that has been quite successful, and it helped us and other platform and partners to bring additional funding to the project, right? So there are challenges even working with academics, in other countries. So it's kind of in some ways similar to industry, but it's just easier from kind of endorsement perspective. I don't know if you want to talk about the Matchmaker.
Yeah, I mean, I think it's kind of similar. Like with Matchmaker, I think one of the challenges, basically BCH has a relationship, a data sharing relationship effectively with each individual node in the Matchmaker exchange network, of which there are about 15. And so and this is fundamental to how Matchmaker works, is each institution forges their own one-to-one relationship, which enables them to share data to say, oh, I have a patient that has a variant in this gene and this phenotype, and then reach out to one another to potentially fuel collaboration on those specific research.
I mean, the advantage, again, to David's point, like seeing 200 physicians, right? Like when the data is in our database, automatically, especially through IPCHIP, which was on the She focus on epilepsy, right? You have many more people really looking at your data, and it's much more likely that the, particular scientist with expertise in your very gene, maybe, who already studies mechanistic models, et cetera, will actually look at your data without even you asking, right? Just kind of a number of eyes on the data and how we scrutinize it through those collaboration expand dramatically. And, we don't even always appreciate how far this goes, right? We often have surprises, completely random places, new findings, and new modes of analysis.
Yeah, and it also can bump a variant in a patient that would otherwise be considered, maybe research. It's a potential candidate. It could be connected. The researcher really feels strongly about it, but there's no other patient in the world that has the same combination of phenotype and type of variant in a gene. And so having these kinds of academic collaborations where we can identify another researcher who has another patient enrolled with the same phenotype and the same gene, that can really, that can turn into real knowledge about the interrelationship between.
The fun project with it, we try to generate gene lists for every single disease. We have the gene list, right? And that's very even controversial because everybody has different definitions, and especially people between different countries, right? In Australia, what potentially causes epilepsy is different than in UK and in the US. And trying to put it together, trying to come up with consensus, and ask the questions, why did you add it to epilepsy? And it just kind of like really extends how far we can take the data.
Yeah, that's interesting. In our last few minutes of our discussion, I want to talk about the future of CRDC and kind of where you see the collaborative going in the next few years. What would you say, so in your work with the CRDC so far, what would you say have been sort of the biggest lessons learned?
I mean, it can be done, right? So this collaborative environment we created, it is effective, it's cost effective, it brings more people to collaboration and more this diagnostic odyssey, it's converging, definitely faster than we've seen it without genomic data. We're building community. I think all of that is very, very positive. This trajectory to cures, it's slow. It's happening. I think in particular, when we talk about industry collaboration, I think there is many opportunities to engage with industry to kind of progress from the being able to identify the gene. We have all the faculty almost at Harvard, between Harvard Medical School Children's. We have many people who work on the mechanism, right? We don't have that many people who work on therapeutics. I think that's, for anybody who's here from the industry, that's really the future of where we should focus. Shira, have you?
I think we're also seeing synergies between the research and the clinical domains,
Right.
which is another, I think, big win as well.
Yeah. So my last question to both of you was going to be, where do you see the CRDC in 5 years or 10 years? Like, what would you say are the key short-term goals, long-term goals? Like, how do you think the initiative is going to change in the next few years?
There are a lot more patients, I think, who need this kind of in-depth analysis. And I mean that both in terms of there are more patients who would benefit from an exome or a genome, kind of more standard genetic testing. But there are also a lot of patients who are going to benefit from the emerging technologies that are forthcoming, right? Like long-read sequencing, reanalysis has been shown to be really effective diagnostically. So I think that's one major push in the future.
Yeah, I think, additional thing, like more engagement with patient families, right? I think novel, especially with the current, funding climate, I think we're going to develop new models to fund the development of cure and therapeutics for rare diseases, and there's with people in Boston, et cetera, there is some new ideas, proposals how to do it. It's still in early stages, but I think that'll expand. We talked about integration with EHR. I mean, the data we're collecting right now is mostly stays in the research domain. And again, to the prior discussion, I mean, hopefully those ICD-10, 11 codes will be increasingly more prevalent and entered correctly to kind of help people when they see their doctors to kind of go in the right trajectory.
And I mean, in the longer term, right? Like people like David shouldn't have problems with, this gene is in the EHR, right? And that should trigger doctors to see certain things. So, I mean, but at the end of the day, the forefront is always going to be in research. No matter how advanced the electronic health record becomes, there's always going to be something new and that's relevant, I think, from the research domain. So.
And Boston, we just converted to Epic last summer. This was a big go live for us last summer, right? June 1st, right? And we were one of the first pediatric institutions to fully embed the genomic module. We're still waiting to kind of tune it up, right? But now the information, once it's confirmed, we actually got for any clinical patients, we got the raw data come back to our research repository, and we can do a deep dive on this research data. If we have any further finding, or if we reanalyze it a year later, we can just call up GDX and have the new report sent back directly and put in your clinical record through Epic, right? So we kind of really trying to push it to where the technology is today and will be tomorrow. So that's maybe even sooner than 5 years, but it's happening.
Great. So I think we have about 2 minutes left. Are there any questions from the audience for Shira and Piotr?
I would like to ask about the connection between the genotype, genomics, and the phenotype for patients, if you have a database for that. And the follow-up question, if someone else is coming from another institute or a doctor with a family from outside, and you would like to use your information for their child, is it possible really to come and use the data or the databases that you have already for specific disease?
So basically, I mean, yes and yes. I mean, for the phenotypic, genotypic data, because we only work with physician scientists who already have the cohorts on a particular disease and they have the registries of phenotypic data. All they were missing is genomic data, right? So we adding to it, we're combining it with the EHR. So internally within BCH, I think this is pretty deep repository. Now we including other MRI modalities that we can bring into the analysis. On the second one is mostly through collaboration. So we wouldn't just kind of give out the data, still pretty strictly regulated. But if you have another scientist somewhere else who want to collaborate with us, I mean, those things happen on a regular basis.
Hey, what are your thoughts on the genetic testing that people can get outside of their hospital, like 23andMe or things like that, where you can just go buy it and send it in?
So if you have genetic testing and you've gotten it from outside of the hospital, we can integrate that data in. 23andMe is not as comprehensive as like an exome or a genome, right? It's very focused. I don't know how much rare disease diagnosis you're going to get off of a 23andMe test, just because of the underlying technology.
Because we harmonize data across, like everything was pretty much GeneDx with the same technology, it kind of gives us much better quality data, especially when we use some of the approaches like from Jason Flanick, right? So the proposal, it was a proposal to basically being there, but it's time consuming cost, you know. So the question is, at the end of the day, if you count everything, is it cheaper for us to just resequence or bring in other data and try to kind of do a one-off to have it integrated? But it's a lot of discussion on the very topic.
Just as a follow-up to that, what are your thoughts on more comprehensive newborn screening program? I've done my research history is at the Broad, where we were part of the Baby Seek program, and that could have been a way that so many more of these cases could have been caught earlier. What are your thoughts on that?
I mean, when our physician in chief, Wendy Chung, I mean, she's basically the leader of this. Wendy Chung, our physician in chief, she's the leader of the.
Oh, no, I mean a newborn screening.
Newborn screening. She's the leader of the newborn.
Like all babies.
Yeah, I mean, I think people are supportive. It's just the question of funding, right? Funding and then analysis. Who's going to analyze that data? So I think 5-10 years it will happen. I think we all supportive of that. It's just a matter of putting it together.
Yeah, I mean the price has dropped significantly. I mean, a clinical genome now is in the hundreds of dollars. So
yeah, and also it's kind of a question of Brigham's end. We don't have newborns at BCH, but on children, right? So yeah, but in general, people are supportive, and there are efforts in that space in parallel to what we do with rare disease sequencing.
All right. Thank you so much, Piotr and Shira.
I mean, just maybe to add to that, the challenge is not just like you can do whole exome, right? But like increasingly finding out it's not just whole exome. For the negative cases, the whole genome along with sequencing. So we're still trying to kind of put together the best pipeline, and this will continue evolving. So the question is kind of what do you offer out of gate for everybody versus how deep you're going to go for the cases when you don't get an obvious answer.
All right, thank you both very much. I think we're unfortunately out of time, but a lot of great questions, a lot of great insight. Thank you, both of you. I am going to turn it back over to Julie to move us forward.
Well, thanks so much for a great panel.